Assalamualaikum and hi everyone. I am Natasha and my team and I will be presenting on molecular techniques in conserving Rafflesia species. Let's begin with an introduction to Rafflesia. Alright, the name of Rafflesia comes from its genus and it is very famous for having one of the largest flowers in the world, often called the corpse flower due to its strong odor. This unique plant is endemic to Southeast Asia but is critically endangered due to habitat loss and climate change. Conserving it requires innovative approaches, and that's where the molecular techniques comes in. There are several molecular techniques used in these conservation efforts. These include DNA barcoding, eDNA monitoring, microsatellite analysis, RNA sequencing, and whole genome sequencing. Each of these methods provides valuable insights into the Rafflesia's genetic makeup and biology, helping the scientists create an effective strategies for its preservation. Now, let's dive into each one of the techniques.
Hi, my name is Nova Najwa binti Muhammad Kasmira, and today I will be talking about a molecular technique used to conserve Rafflesia. First of all, we have DNA barcoding. DNA barcoding is a method that helps us identify exactly which species of Rafflesia we are working with, so we can better understand and we can protect them. Scientists often use DNA barcoding to identify species, and here we focus on two important regions of the chloroplast DNA, which the first one is motilus K and the second one is Ribulus 1,5-biphosphate carboxylase. These regions are like genetic IDs for plants. Now, what's really interesting about Rafflesia is that in some species like Arlagaceae, scientists like Molina in 2014 found almost no plastic DNA at all. This makes it harder to classify the species based only traditional traits. So that's why we are applying DNA barcoding steps. By looking at these specific DNA regions, the researchers can still confirm the species even in fragmented habitats or when the plants are mixed up with others. This technique is important especially for identifying cryptid species, which those that look alike but are actually different. And with some species of Rafflesia potentially losing their plastic genomes over time, DNA barcoding is a game changer to make sure we are identifying correctly. It helps track the species present in habitats where they might be in danger or hard to find. So to wrap it up, DNA barcoding is a super important tool for figuring out which Rafflesia species we have, even when they don't look like typical plants.
Did you know one of the world's largest and rarest flowers, the Rafflesia, is at risk of disappearing forever? Known for its massive blooms and unique ecology, this incredible plant is threatened by habitat loss. But scientists are using a groundbreaking technique called environmental DNA or eDNA to give it a fighting chance. eDNA allows scientists to detect traces of genetic material left behind in soil or water like a biological fingerprint. For Rafflesia, this means researchers can locate these plants without disturbing their fragile environment or waiting for them to bloom. Here's a figure from a Satanuncool study. It shows how many cycles of qPCR were needed to detect eDNA in soil samples. Fewer cycles mean more DNA is present. The closer the soil sample is to the Rafflesia, the stronger the DNA signal. This proves how effective eDNA is for monitoring this rare species. Rafflesia doesn't bloom often, making it hard to find. But with eDNA, scientists can detect these plants year-round, helping conservationists map their habitats and protect them from destruction, all without harming the plants or its ecosystem. eDNA is revolutionized conservation, offering new ways to protect biodiversity. By supporting this effort, we can help incredible species like the Rafflesia thrive from generation to come. Let's work together to preserve our natural world.
Now, let's move on to the third technique, which is the microsatellite analysis. So, microsatellite analysis is a powerful technique widely used in conservation genetics. Microsatellites, or short tandem repeats (STR), are repetitive sequence of DNA that vary between individuals. These markers are ideal for studying genetic diversity and population structure in endangered species such as Rafflesia. In this process, DNA is first extracted from the Rafflesia tissue samples. The specific microsatellite region are then amplified using polymerase chain reaction, or we call it as PCR. The amplified DNA is analyzed to determine the number of repeats, which reveal the genetic variation. This is proven by a study by Pelser in 2017, which found a good amount of genetic diversity in Rafflesia lagaske in Philippines. Many different genetic variation or allele were present across the population studied. This indicates that these plants have a healthy mix of genes, which is essential for their survival. Using microsatellites, scientists can identify which population are genetically healthy and which are at risk of inbreeding. This information is vital for developing effective conservation strategies to protect these rare plants. Microsatellite analysis give us the tools to understand the genetic health of Rafflesia population and to ensure their continued existence in our ecosystem.
Hi, my name is Nur Indina Binti Ahmad. For the next molecular technique, I will explain about whole genome sequencing that is applied to have a better understanding about Rafflesia parasitic relationship. Molecular techniques like whole genome sequencing have revolutionized our understanding of parasitic plant, especially fascinating species like Rafflesia, that we all know as corpse flower. Rafflesia depends entirely on its host plant, typically Tetrastigma muffins, for survival as it lacks roots, stems, or leaves. This unique parasitic lifestyle makes conservation efforts particularly challenging. By sequencing Rafflesia genome, we can identify the specific genes that enable its parasitism. For instance, genes that facilitate nutrient absorption, suppress host defenses, and ensure host recognition are crucial for the plant's survival. Interestingly, Rafflesia has also integrated genetic material from its host over millennia, a process known as horizontal gene transfer. Hence, this makes its genome a fascinating archive of host-parasite evolution. The importance of this research extends to habitat management. By understanding the genetic basis of Rafflesia host dependency, we can prioritize the conservation of its host plants and ecosystem. So, protecting Rafflesia is not just about saving a single species, but it is also about maintaining the intricate balance of tropical forest ecosystem. Hence, this knowledge derived from advanced molecular tools provides a roadmap for preserving these extraordinary parasitic plants for future generations. And I will pass the next molecular techniques to my friend.
Hi, my name is Nurin Najwa and I will explain on the last molecular technique for conserving Rafflesia, which is RNA sequencing. RNA sequencing allows for the comprehensive analysis of gene expression and the identification of regulatory RNAs including microRNAs and long non-coding RNAs, which is for plant development and adaptation. And according to Amini and Associate 2019, in the context of Rafflesia, RNA sequence can be employed to characterize the microRNA's transcriptome that reveals both conserved and species-specific microRNAs that may play important roles in the unique developmental processes of this genus. The analysis of microRNAs facilitates the understanding of how Rafflesia interact with its host plants, particularly Tetrastigma, from which it derives nutrient. In RNA sequence, the long non-coding RNAs can uncover the evolutionary significance and functional diversity in Rafflesia. Besides, the application of high-throughput sequencing technologies enables the detection of novel RNA species that may not be conserved across species. By integrating RNA sequence data with computational analysis, researchers can predict the target of identified microRNAs and long non-coding RNAs, thereby elucidating their roles in the complex regulatory networks that underpin Rafflesia development and its parasitic lifestyle. To sum up on this, RNA sequencing presents a powerful molecular technique for conserving Rafflesia by providing a detailed understanding of its transcriptomic landscape.
In conclusion, we have discussed the urgent need for Rafflesia conservation due to its ecological importance and the threat it faces. Utilizing advanced molecular technique allows us to identify and monitor these species effectively. Techniques like DNA barcoding help distinguish between species, while environmental DNA analysis provide a non-invasive way to locate them. By understanding their genetic diversity and exploring their unique biology with whole genome and RNA sequencing, we can develop targeted conservation strategies. Together, these efforts will ensure the survival of Rafflesia and the health of its rainforest habitat. And that is all from us. Thank you.